Okay, let's pick up where I left off here. So when we think about the flow of energy through an ecosystem or through anything, it involves the activities of the chloroplast and the mitochondria for what we're going to be talking about. True, it starts with the sun, but without those chloroplasts carrying out photosynthesis, we wouldn't be able to convert that solar energy into carbohydrates, right? And hopefully this is having a flashback for what we talked about in weeks prior. Cellular respiration is the coupled reaction where we take those carbohydrates that are created from photosynthesis and use the energy stored in that carbohydrate to build ATP, charging up those batteries from ADP to ATP. So again, thinking about how the organelles that we learned about in the last chapter work with the flow of energy. Here's a nice little graphic where we have the chloroplast up here. The solar energy penetrates through different layers of the cell and the tissue of the leaves and the stem and activates the little thylakoid membranes that we talked about. And of course, heat is released, but some of that energy is used to be converted into a type of chemical energy, because we know that carbohydrates are chemicals. And then that chemical energy can be broken down by the mitochondria. Sure enough, some of it is lost as heat, but some of that energy that's stored in the carbohydrate is charged up those ATP batteries. Okay, and of course, you've probably learned this in middle school and high school, is this interchange of chloroplasts need carbon dioxide and water to do photosynthesis, and they release oxygen. And sure enough, the mitochondria need oxygen to run cellular respiration, and they release carbon dioxide and water. If you go back to your notes from the last lecture, you'll see those reactions, so you might copy them back down. That ATP that is made in the mitochondria can be used for all sorts of different activities for the cell, chemical work of changing one chemical into another. They can move molecules in and out of the cell. We talked about that there's those proteins in the cell membrane that are used for transport, and there's other ways that we'll get to in just a minute. And also mechanical work. We can get things to move, like the flagella, like our the filaments of cytoskeleton that we talked about. And of course, if we think about it at the organism level, not just the cellular level, we have humans that are involved in cycling these molecules between plants and animals and one type of animal to another. We're part of that cycle as we think about the energy flowing from the sun to the plants into various levels of animals. We inhale oxygen. We get our chemicals from the plants and animals that we eat. And some foods allow us to create or produce that ATP that's required. And of course, when we think of those calories, there's some foods that have a high amount of calories in them, which means that we can produce more ATP from them. It does not mean that they're healthier. I don't want to make it sound like that. I'm not saying that all calories are the same when it comes to your health, but when it comes to the scientific reasoning behind calories, they are the same. So, thinking about this, kind of taking a time out here. I was hoping to get this far through the first lecture and didn't quite make it. So go back in the notes, make sure you know what is energy. What is the definition in science for energy? Tell me what a calorie is. Now, again, I'm not going to worry about kilocalorie versus calorie. You should know what kind of unit of measurement it is. What does conservation of energy mean? What does that law mean? How does entropy affect life? And again, this is just a big picture of that energy is going to start in a really useful form coming from the sun. We can use it in a lot of different ways, but then after being converted from one to another, to another, to another, it becomes less organized. And in order to get it in an organized form, sometimes it must have energy input. I need you to have a good understanding about what ATP and ADP are. True, you might need to know the name, but that's not really the main idea. The main idea is the idea of how those molecules are used in the cell for energy and how one gets changed to the other. And I want to make sure you know how chloroplasts and mitochondria are involved. So again, you might pause right now and be able to fill those in with your notes by going back into the lecture slides or back into the video to find out more. Another cell process that we need to talk about is how enzymes work. Okay, so enzymes are a type of molecule. In fact, most of them are proteins. Okay, and what they do is they help metabolism. They may help build, take two molecules and pull them together, or they might take a molecule and help break it down. So the picture here, the blue thing here is this enzyme. It's a big globular protein. We already talked about this, that, proteins are made on the ribosomes. We put together that string of amino acids based on the code of DNA. And then that string of amino acids go into the rough ER and they get modified and changed, and their shape becomes something that's very important to their structure, right? And the Golgi might help out as well. So here's a specific example of a protein that its shape is super important because there's a spot, cleft, an active site where a substrate, or in a chemical reaction we would call these the reactants, come in, and that enzyme helps break it apart. Now, I do want to say that there's some enzymes that do the reverse, right? They take two parts and then they help to bond them together. So the things, the molecule or molecules that an enzyme acts upon are called substrates, and they could facilitate in breaking down molecules or putting them together, which is what synthesis is. Most of them are proteins, and essentially this reaction of taking this orange molecule and breaking it down to a red and a yellow, to be general, it would happen on its own, but the enzymes speed it up. Okay? So thinking about enzymes as a protein, you should already click together that, it's made of a string of amino acids from the ribosomes, and its shape is important to its function. Now, this active site is where the substrate actually interacts with the enzyme, and there's kind of two ideas of how this works. The first is the idea that it's like a lock and a key, and this is a good way to describe enzymes because enzymes are very specific to the molecule it works on. So if I go back, this blue enzyme can only work with this orange substrate. If you tried to fit in some other shape, it's just not going to work. So it's the idea of a lock and key. This enzyme does not work with any other substrate. However, there's another idea when it comes to enzymes, and they kind of go together here, but the induced fit model means that when the substrate and enzyme comes together, it actually undergoes a change slightly in its shape so that the substrate is a perfect fit. That we actually are finding out now that while it's kind of a one-to-one relationship between enzymes and substrates, the enzyme can actually be a little moldable for the substrate. So if the substrate's a little different, the enzyme can still work. The thing that's important to note is that enzymes are never part of the actual reaction. Like if you saw a chemical reaction, let's say photosynthesis, there's actually enzymes helping out that entire process along the way, but we don't write it in the chemical reaction because it's just helping the molecules come together or be broken apart. So here's kind of a side-by-side idea. So here's an idea of the lock and key model where these two substrates are just a perfect fit. Nothing else would fit in there, and that enzyme helps to lock it together into one. So this is the opposite, where this would be a synthesis reaction, right? Two parts coming together to be one. The induced fit model, and it's not one or the other, these both kind of fit together to understand our picture of enzymes, is the idea that if you look at this enzyme there, like that's not going to fit. But when the substrates get there, the enzyme forms around it. And again, the end product is the same. An interesting reaction when it comes to enzymes is inhibition. So sometimes there's other molecules that actually block the active site, and it prevents the substrate from combining with the enzyme. So here's some examples, like penicillin actually works in this way. So penicillin was the first antibiotic that we produced, and at this point it's not very effective with many bacteria, but we have other types of penicillin, like in the same class, like amoxicillin and things like that. And what penicillin and its derivatives do is it blocks a certain bacterial enzyme that builds the cell wall. It blocks it so the cell wall can't be made properly, and so the bacteria dies. There's also feedback inhibition, which means as the enzyme is going through its chemical reaction, either breaking things up or building things together, once there's enough product, the enzyme becomes inactive. Okay, sorry, that should be one word there. It should become inactive. The idea is, hey, we have enough, we don't need more, and so the enzyme becomes dormant until there's more product that's needed to be made. So essentially how an enzyme works, we know what it does. It speeds up a chemical reaction by bringing molecules together or breaking them up, helping that chemical reaction to go faster. But how it actually does that has to do with the energy of activation. So again, this is a little more chemistry than what maybe you're prepared for. But the thing is, in order for a chemical reaction to happen, molecules have to physically bump into one another. And that's kind of hard to do sometimes. We're thinking about life inside the cell where it's mostly water and molecules are kind of floating around, maybe attaching to cytoskeleton to move around. But in order for two that are supposed to bond together to get together, they have to be not only near each other, but in the right orientation. So the activation energy, and there's a little, capital E sub A, that's the energy needed to cause the molecules to react to one another. Okay? And what enzymes do is it lowers the amount of energy required to get the reaction going. Because enzymes bring those substrates into contact and make sure they're in the right position for the reaction to take place. I kind of like to think of enzymes as a matchmaker, right? The chances of you and that special someone running into each other in the 7 billion people we have in the world may seem kind of rare, but sometimes it helps to have a third person say, you really need to meet so-and-so. And they get you in the same physical location to see what reaction occurs. And when it's said and done, that matchmaker should walk away from the relationship and not have any part of it, right? The enzyme does the same thing. The enzyme is a molecule that is not part of the chemical reaction directly, but makes sure that the molecules needed for the reaction are in the right place at the right time. And does that because now the energy needed to get those molecules together is a lot lower. So here's a nice little graph. You can find this a million places. So here are the reactants. Remember, those are the molecules are at the beginning of the chemical reaction. Without the enzyme is this red line. So look at this steep hill, the amount of energy needed. Now once we get to the top of the hill, then it's free fall down. No big deal. And there's our products at the end. So let's start with one molecule, and then we end up with two at the end, let's say. An enzyme, yes, there's still an activation energy, but it's much less needed because the enzyme is doing the work of getting those reactants in the right orientation, and then the same products are at the result. So again, beginning to end is the same with and without the enzyme, but the in-between, there's less energy needed. And again, when we think energy, we think ATP. There's less ATP needed. So a lower hurdle is easier or faster to get over, right? I don't know if any of you have ever run hurdles, but just like trying to jump over anything, the lower the object is, the easier it is. So a lower activation energy makes the reaction happen easier and faster. And again, timeliness is important inside our cells. We need the molecules we need now. The oxygen, the carbon dioxide, the sugar. And so enzymes help everything happen in a timely manner. Okay, so I've got about 10 seconds here. It says, these are the questions I need you to know the answers to. So make sure you go back into the presentation to find those out.